Okay, so we know one is acetylcholine, right? So acetylcholine is our main neurotransmitter, the most abundant in the body. We've also got norepinephrine / epinephrine. They pretty much bind to the same receptors. You can think of them together. And the neurons that release them are the preganglionic neuron releases acetylcholine. In the sympathetic system, the postganglionic neuron releases norepinephrine or epinephrine. Okay, but in the parasympathetic system, both neurons release acetylcholine. In the neuromuscular junction, we also see acetylcholine as well. What is the neuroaffected junction? So this is the specific junction between the postganglionic neuron and the effector cell in the sympathetic, in the autonomic nervous system. So both parasympathetic and sympathetic. And then in what regard does it differ from the ordinary neuron to neuron synapses? In what respects are they the same? So they differ two main things. Let's kind of go back here. Two main things here. So first off is the presence of these swellings called varicosities. The second difference is actually the distance across this cleft. So if we look at this synapse here, this junction, this distance is greater at the neuroaffected junction than it is in a neuron to neuron synapse. And we can think of that as being at the autonomic ganglia, for example. So the distance across the cleft is greater at the neuroaffected junction. There's also the presence of these swellings called varicosities that we don't see anywhere else but the neuroaffected junction. Okay. Yeah, any questions here before we keep going? I'll pull up the other questions here in the meantime. Okay, I think we also skipped over these, which were the final questions for objective 1. So let's look at these quickly. So the first question here, which neurotransmitter is released by motor neurons at the neuromuscular junction, and then to which type of receptor does this neurotransmitter bind? Very, very, very straightforward for the somatic nervous system, right? One neurotransmitter, always and only acetylcholine. One type of receptor, always a nicotinic cholinergic receptor, okay? Fast receptor, ionotropic receptor. Those 4 terms are pretty much mean the same thing here. How many motor neurons innervate a single skeletal muscle fiber? 1, right? 1 motor neuron, 1 muscle fiber. No, that's, sorry, 1 muscle fiber, 1 motor neuron, but 1 motor neuron, many muscle fibers, okay? So that's going to be 1 motor neuron innervating a single skeletal muscle fiber. So let's move on here to objective 2 in the somatic nervous system, which is applying our knowledge of the events at the neuromuscular junction, and then talking about some natural toxins that affect this region. So there are 3 toxins we're going to talk about, and we've got the junction pulled up here for reference. A couple of things I want to point out about the junction. So we call this the motor end plate, and we can see the highly folded nature of skeletal muscle of the motor end plate at the neuromuscular junction. Okay. I also want to point out the presence of a nicotinic cholinergic receptor and the presence of the only neurotransmitter here, acetylcholine, as well as the presence of acetylcholinesterase, which is that degrading enzyme that's seen at all cholinergic junctions, okay? So let's talk about the first toxin. This is called latroxin. This is actually found in the venom of a black widow spider, and it's going to stimulate the release of acetylcholine into the junction, okay? As you would imagine, that's going to just increase the signal transmission at this junction, which will cause rigidity, spasticity, and just this overexcited muscle tissue, which can lead to that type of physical presentation in patients, right? The way that this can become fatal is, again, with the respiratory muscles. If there's rigidity or spasticity, which is like the overexcitement of those muscles, we can see interruption in breathing, okay? That can become fatal that way. So that's latroxin, black widow spider, increases or floods extra acetylcholine into the neuromuscular junction. Let's talk about crotoxin. This is from the venom of a rattlesnake. This is going to have the opposite effect of latroxin. This is going to inhibit or block the release of acetylcholine. The symptoms are going to look a little bit like the next toxin that I'm going to talk about, but we want to understand that the source is different. So blocking the release of acetylcholine into the junction will prevent or, stop this transmission of this signal. That's going to present with the opposite symptoms as what we saw with latroxin. So weakness, paralysis, and that will become fatal in the same way, though. It's going to affect respiratory muscles, which are really important for breathing. And so if we have an interruption of those muscles, we can, that can be fatal in that way. So latroxin, more neurotransmitter, crotoxin, less neurotransmitter, both can become fatal by their involvement with the respiratory muscles. The third toxic compound here is curare. And curare is usually going to have a toxic event. It's actually a poisonous substance that Native Americans would typically put at the end of their darts and shoot these darts to paralyze, animal, prey, and so on. And so it's going to have an effect on the nicotinic cholinergic receptors on the motor end plate. And that is going to, again, block the transmission of this signal and result in paralysis, in weakness, and it can become fatal, again, involving the respiratory muscles. So this is going to have a similar presentation as crotoxin, but the source is different, right? One is blocking the neurotransmitter from entering the junction, not really affecting the receptors, that's crotoxin. Whereas curare is not really affecting the amount of neurotransmitter, but it's blocking the ability of that neurotransmitter to bind effectively to these receptors. And I'll point out here, curare is very similar to myasthenia gravis. I've been talking about myasthenia gravis. This is, again, altogether a different source, but pretty much the same end result. This is an autoimmune attack on these receptors. And so these receptors are injured, are damaged, are removed, are less effective. And so this would also present the same way that curare would, with paralysis, with muscle weakness, and then respiratory disturbances. Very interestingly enough, the treatment for curare, and if you go back to that table that we talked about with all the, the pharmacological uses of some of these autonomic receptors, and we look at the treatment for myasthenia gravis, it's really blocking acetylcholinesterase. So it's an ACEI, an acetylcholinesterase inhibitor that blocks this kind of Pac-Man enzyme that we've been talking about that degrades the excess neurotransmitter. And we can kind of see how that would make sense, right? If I have trouble with binding of neurotransmitter to this receptor and the transmission of that signal, then it would make sense for me to block this enzyme and allow that neurotransmitter to linger at the junction for just a little bit longer than it would normally in order to increase any chance of binding of whatever receptors are existing or are working to whatever degree, and just try to kind of boost that signal a little bit more. So interestingly, when we give these myasthenia gravis patients this inhibitor of acetylcholinesterase, they turn out tremendously. They see tremendous improvements. They get stronger from that muscle weakness that they, were presenting with. So it's really amazing how we can take our knowledge of this junction, of the players at this junction, and we can block some of these structures and players to bring about desired effects. And we can also use our knowledge of the receptors to treat certain disorders as well. Okay. I'll pause here, see what questions you may have had on that second objective with the toxins. Otherwise, there's this study assignment that I think will be very helpful for you all this weekend. So I will encourage you to draw out and compare the following efferent neurons and the following synaptic events. So the neuromuscular junction, right? You're going to draw out the motor neuron, where it arises from in the spinal cord, what the synapse looks like, what the neurotransmitter involved, what the neurotransmitters are, or is in this case, what the receptor is. Also do that for the parasympathetic system. So those two neurons, where they arise, the length, how they differ, and then do the same thing for the sympathetic series of events, okay? It's really important for you to now be synthesizing, right? Taking all of this information, comparing, contrasting, how does this differ from that? you know, like just being able to fill in those small gaps. That's what's going to get you into the upper third on this next exam. Having an overall good, clear picture of how all of these events differ, how they're alike, okay? So a really quick example off the top of my head here, I can ask you, will I see a nicotinic receptor at a neuroeffector junction, right? You should have a quick answer to that. The answer is no. You only see nicotinic receptors at the neuromuscular junction or at the neuron-to-neuron synapse in the autonomic system, right? So make sure you have a good understanding of some of those functional differences, structural differences in these three divisions here. Okay. So let's look at a few checkpoint questions, or not checkpoint, poll questions here, multiple choice questions. I'm gonna launch a poll and give you a minute or so to think about an answer and poll in. Okay, final couple seconds here, 5 or so seconds. All right, let's take a look here. So kind of an even split among these responses. So let's look at, but there was an overwhelming response for D. So let's look at the question, which is not true about adrenergic receptors. So alpha-1 and alpha-2 receptors are generally excitatory, that's true, have a greater affinity for norepinephrine than epinephrine, that's a true statement. Beta-1 and beta-3 receptors tend to be excitatory, also true, have an equal affinity for norepinephrine and epinephrine, also true. Beta-2 receptors are generally inhibitory, true, and have a much greater affinity for epinephrine than norepinephrine, also true. A, B, and C are true. Alpha-1 and alpha-2 receptors are generally inhibitory. That's not true, right? Alpha-1 and alpha-2 receptors are generally excitatory and have a greater affinity for epinephrine than norepinephrine. That's also not true. Alpha receptors in general have a greater affinity for norepinephrine than for epinephrine. Okay, so D was completely incorrect. Okay, let's look at a couple others here. So the answer here was D as the incorrect statement. Let's look at another question. So I'm going to launch a poll here again. All right, last 3 or so seconds here. Okay. All right, so we had an overwhelming response for A. So the question asks, in the parasympathetic nervous system, which neurotransmitters are released from the preganglionic and postganglionic neurons? So really important here, parasympathetic system, okay? And let's look at the options. So A says preganglionic neurons release acetylcholine, postganglionic neurons release epinephrine or norepinephrine. And that's what most persons selected. That is not correct, okay? What system is this for? What system is a representative of? Or what division, rather? This is the sympathetic, right? So that is not correct here because we're asking about the parasympathetic. Let's look at B. Preganglionic, epinephrine, postganglionic, acetylcholine. That's not even a correct, that's not even possible, right? That doesn't happen. Let's look at C. Preganglionic acetylcholine, postganglionic acetylcholine. That's correct, okay? And then obviously D is going to also be incorrect. So this is asking about the parasympathetic system. And I keep stressing here, the parasympathetic system, both neurons release acetylcholine, okay? Preganglionic neurons, postganglionic neurons, they both release acetylcholine. In the sympathetic division, okay, preganglionic neurons release acetylcholine, but postganglionic neurons release norepinephrine and epinephrine with the small exception of sweat glands, okay? So the correct answer here should have been C. This is the kind of detail that you really need to get comfortable with. So again, I encourage you to draw this out, really look at each of the divisions, really compare and contrast the differences and anatomically, functionally, which receptors are where, which neurotransmitters are where, etc. Let's look at another question here. Okay, last 10 seconds. All right, let's take a look here. So a lot of response for D. The question asks, after a neural signal, sodium flows into a muscle fiber and produces a depolarization called an end plate potential. That is similar in many ways to the excitatory postsynaptic potentials. How is it different? Okay. End plate potentials can sum together. No, they cannot. Okay. Hormones can also cause muscle contraction. No, it cannot. Whereas we saw that norepinephrine and epinephrine could bind to an adrenergic receptor and bring about muscle response, those hormones are not found at the neuromuscular junction. Okay, so they cannot cause muscle contraction. It must be a neurotransmitter, acetylcholine. End plate potentials are excitatory. End plate potentials are excitatory, but so are EPSPs. Okay, so that's not a difference. And then the fourth option here, end plate potentials normally have enough amplitude to contract the muscle. And that's correct, okay? And that's, I think, yes, that's what most response was for D. So the important difference here is EPSPs can get us closer to depolarizing the membrane, to generating another action potential, right? Remember, we said that EPSPs are summed and analyzed, and then we would determine if we meet that threshold. With muscle, with skeletal muscle, that is not the case, right? It's always excitatory and it's always enough amplitude to actually bring about that end plate potential, that muscle contraction, okay? Always excitatory, always going to generate muscle contraction or an end plate potential. And they cannot sum either, right? The way that we see that those graded EPSPs can. So the answer here would have been D. And then let's look at a fourth and final question here. Oops, I did not, did I? Oh, I did. All right, last couple seconds here. All right, let's take a look here. So which is not an anatomical arrangement for the sympathetic signal from its point of origin to effectors? And we had a lot of response for C, which says synapse on the sympathetic chain, then adrenal. Okay, so that is not correct. So this would have been the correct answer. Let's look at some of the others. Synapse at the sympathetic chain, then postganglionic neuron synapses on the organ. That's true, that's a possibility. Synapse on the collateral ganglia, then to the effector organ, also a possibility. And then D is onto the adrenal medulla, so that's also a possibility. So C is not an option. It doesn't synapse onto the sympathetic chain and then the adrenal gland, it's either or. So it's one of those three routes of communication and not, two out of those three routes. So it has to be one of those three routes. Okay. All right, and so the correct answer